Thank you for joining us for another episode of Biotech and Life Sciences CEO Stories. Today, we're pleased to have as our guest Bill Korinek. He is the co-founder and CEO of Astrocyte Pharmaceuticals, a company developing neuroprotective therapies for treatment of stroke, traumatic brain injury, and even concussion. Bill, maybe you would like to take a minute or two and give us a little of your background on your path before astrocyte.
Oh, good. Tim, thank you for the introduction and for having me on the podcast. Yeah, looking forward to the discussion. For myself, my background is I started off as a scientist. I always loved science, loved learning about molecular biology, cellular biology, the magic of DNA. So I did an undergraduate major in both biology and chemistry. And from there, I went directly to graduate school. I went to Harvard and got my PhD in molecular and cellular biology. And it was an exciting time. It's when the genome was being sequenced and
Right.
lots of companies were popping up. Everybody's talking about DNA and genomes. But I probably did a little bit of a left turn after graduate school. A lot of people get their PhD and then go directly to, postdocs or academic positions or into companies. I went into consulting. I went into a management consulting firm by the name of LEK Consulting. So it's a well-known strategy consulting firm, management consulting. What was nice about LEK at the time is they had a very large and growing life sciences practice. So I spent 5 years at LEK at a time when companies like Biogen and Genzyme and Millennium Pharmaceuticals were all kind of really growing and getting off the ground further and exploiting the genomic era. And so I worked at LEK for 5 years, but worked for about 40 different companies during that time, almost all biotech, some small, some large, some large pharmaceutical companies, a couple medical device companies, retirement home at one point, but really got to see
Okay.
a pretty broad spectrum of how this industry works and understand that landscape. Understand a lot of the strategies and the players and just the competitive nature that exists. And then with that broad landscape, I decided to take a chance and go into a larger pharmaceutical company, and I joined Pfizer and was in their strategic kind of group, basically that developed and kind of drove a R&D strategy. So I was there for about 10 years and helped guide our various research units into whatever the therapeutic areas that were of highest priority. What's our 10-year plan for getting more from a small molecule driven company to being more in biologics with not only antibodies, but what we'd be doing in RNA and cell therapies and everything else over time. And yeah, so I feel like I got to be going from the strategic to learn more of the operational side of things. And it was nice to have that, scientific foundation, but really also get the business side of things during that time frame as well.
Right.
And that kind of eventually led me to the opportunity to Astrocyte Pharmaceuticals, which I'm sure we'll talk more about.
Sure. How did you meet your co-founder, Dr. Lechleiter? And what inspired you to start astrocyte?
Yeah, so I've known Dr. James Lechleiter for forever. He's a family friend, and actually, I got to know him or actually visit his research lab back when I was in high school. He was working at the Mayo Clinic at the time, and I had the opportunity to visit him, see what research he was doing. And I think it's some of those early interactions with him and his wife, Patricia Camacho, also a scientist, who really opened my eyes to the world of DNA, recombinant DNA, cloning, the broad potential of this field. And so it actually inspired me to I think, go more into the biochemistry world. And we've, been friends through the years. And it was just kind of out of the blue when he contacted me in 2014 and, just told me that his current lab at University of Texas Health at San Antonio had just received a patent for some neuroprotective molecules. And I thought he was working on some earlier research, not actual potential drugs. And he had asked me if Pfizer might be interested in licensing these compounds or doing a collaboration. And I had to tell him, unfortunately, that the answer would be no. That his
Okay.
research, it was just, it's too early. Some great, exciting results here in mice, but it's too early for Pfizer to be interested. But Jim, you should form a company. This is an exciting area. It's a huge opportunity, and there's nothing really in neuroprotection. These are, large indications, huge unmet need, and there's so much you can do now, with contracting out manufacturing and safety and all the studies you need to do. Jim, you should form a company. And he was an academic, said that not something he knew how to do. So we kept talking, and 4 months later, we were founding Astrocyte Pharmaceuticals.
So it's interesting. Bill, one of the missions of this podcast, as I see it anyway, is to inspire and encourage other founders, and especially what I might call pre-founders. So my question is, you meet Dr. Lechleiter, or you've been a family friend, but you hear about his work, and it's just a little too early for Pfizer. So ultimately, you decide to leave this stable, I assume well-salaried, kind of nurturing corporate environment at one of the most prestigious pharma companies in the world for a startup. Why?
So great question. I definitely, went back and forth, kind of multiple times on whether this was something I should do or not. And I think we went back and forth in terms of, how we would, fund it and what would be the priorities, coming up with a business plan. And it really just kind of got to the point where I knew that when work was getting, ramping up, I'm part of the, doing the budget cycle at Pfizer, and I knew I'd have to start giving 110% of my, effort and time to Pfizer, or 110% to this new opportunity, that would be Astrocyte. And I needed to make a choice. And as I kind of weighed the options, one thing that I thought was a fairly positive, good argument was that if I do this for 2 years, and even if the science doesn't pan out and it fails, I think it will have been an enriching experience. That it's something that I will have learned, I will have developed. It kind of might be a little bit like getting an MBA in a sense. I think I would have, learned something from the experience, and I would be a better, employee, colleague at whatever future job I might do. So I didn't think I was going to lose 2 years. I was enriching myself over those 2 years, and then probably most importantly, I just knew that I would regret not doing it. It's one of those things. This looked like just a tremendous opportunity. It probably wouldn't go forward. Dr. Lechleiter would probably not, start this if I did not get involved. And I think I would just kick myself, multiple years down the road if I just never tested this and found out if it worked or not. So that was probably the final push to say, let's do this. Let's see how it can work.
What about in terms of, let's say, your own financial system or situation, excuse me, personally? Were there sacrifices you had to make? I don't know if you're married, but did you get some pushback from members of the family or friends? What was it like? What was
Yeah.
that thought process like?
So I am happily married, and I'm lucky to have a very supportive wife, Christina, and 3 young kids at the time. And it is critically important to have a good support network if you're going into a startup situation. And I was fortunate where my wife was very supportive in terms of what we, would be doing here, both financially and time wise. If you're jumping into a startup environment, not only is usually the pay much less and maybe more unpredictable, but it's also the time commitment. You basically are, spending, 60, 70, 80 hours or traveling and doing a lot more stuff that makes it a little more challenging for, what needs to be done with, the children and other, family relationships, etc. So yes, I was very, lucky and fortunate to have that supportive wife who we thought we would be doing this opportunity for a couple of years before it kind of took off. And it's now been 10, so it's gone on much longer than her or I thought so at the beginning. So again, so even more important to have that supportive person involved. But yeah, it's definitely a challenging environment. And if you're not prepared for that, it can be, make you give up pretty quickly. I know there's been a couple times during the course of our company where we had to pause salaries, essentially for kind of a couple of the key people. So if you don't have that support network, it's going to be a long road.
Did you make financial arrangements anticipating those kinds of challenges, or was that something you were in a position to be able to do, or?
So we basically had a kind of enough of a little bit of a nest eggs, up so that we could not only kind of put that into the company, but knew we had a cushion for a while. And yes, I think you kind of need to have something like that. Some people are maybe might be fortunate too, where you can kind of continue in your old job or do something in parallel to the funding and things take off. For me, I needed a clean break. You can't be working at a company like Pfizer and then having another biotech company on the side. You need to draw a line there on what's owned by who. But maybe other people can actually be in a little bit more of a venture situation where they can have a couple different opportunities that can balance things out, as things move forward. But it's something you need to plan for.
Okay. You mentioned Dr. Lechleiter's approach was novel, and that he was targeting astrocytes when historically maybe most of the research had been focused on the neurons that transmit electrical impulses throughout the body. Why do you think that your approach with astrocytes was previously overlooked?
Yeah, that was probably one of the most intriguing things about his research and why I was quite excited from the very beginning. For the last 100+ years, neuroscience has been dominated by the study of neurons. That's how it gets its name, neuroscience. Everything's the central nervous system, everything's neuron driven. And from the early days, people could, stimulate one end of a neuron and see that electrical signal travel down. So everybody's just thought of that as the most important part of the brain. And the glia cells, the other cells in the brain, have always just been called they're the glue. Basically, people thought of them as a structural support, bystanders, gap fillers, not important. They're not transmitting signals or anything. So really, the research on the glia cells has really lagged behind, and only probably in the last 30 years has it really started to ramp up. It's still dwarfed by what's done on the neuron side of the cells of the brain, but it's the most common cell in the brain. Astrocytes are the most common glia cell, the most common cell. They are highly conserved throughout evolution. And if I were to, digress, there's a interesting anecdote. Albert Einstein, famous scientist that we all know and love, uh, after he died, his brain was preserved, and people studied his brain. They looked at his brain from a lot of different angles, and tried to figure out was there something different about this, genius, and they could not find anything for a long time. It was, the brain was the same size, same weight, same general makeup. We looked at the neurons. There's the same number of neurons, same density of neurons in his brain. So nothing too significant until Dr. Marion Diamond at UC Berkeley looked at this very closely. And in her research, one thing she found was that there were more glia cells. There were more astrocytes. It was more dense in terms of the number of astrocytes in his brain. There's like twice as many glia cells in part of the brain as parietal cortex, the part that's dealing with abstract thinking and complex thinking. So there's been hints over the years that astrocytes are very, exciting, correlated with a higher function, higher survival in some other areas. There's a myriad of functions associated with them. So for me to know that this was not just the same old approach to trying to change neuronal chemistry and synaptic transmission, it was a different approach using the support cells, made it very exciting. So that was part of the reason that we dove in.
Can you give us kind of a high level description of astrocytes and what their critical function is in terms of being caretaker cells in the brain, and why targeting them holds promise?
Yeah, so the astrocytes are they do have, like I said, a myriad of functions, but one of the more common ones is just as a caretaker of various environments within the brain. They actually have a kind of a tiled nature to them, where they are spread out over space, and each astrocyte kind of almost monitors a specific area of the brain with that tiled approach, and they touch every other cell in the brain. They touch every synapse that is formed between neurons. They touch and form the blood-brain barrier. So they really are the sentinel cell And they have, again, a myriad of functions. They really, almost every kind of amino acid, every neurotransmitter exists as a shuttle between astrocytes and neurons. They really are involved in a lot of the care and feeding, essentially, of those neurons and maintaining a kind of a homeostatic environment. And I think an analogy that we like to use is that the astrocytes are like the parents, parent cells in the brain. Neurons are like kids. And when you think about the kids getting into trouble, like there might be during an injury or stress situation, the neurons act like kids. They actually start firing their signal more. They go a little bit more crazy and they deplete their resources. They tire themselves out. They have these recurring depolarizations. And then actually can end up, if it's serious enough, it actually would cause those neurons to lyse and die. Um, but it's the astrocytes that will care for them and actually try and like clear these neurotransmitters. It's glutamate. They'll provide more kind of other resources to these cells that can actually be oxygen or you know energy substrates to the neurons. So again, it's like parents that are trying to calm down the kids and calm down that part of the environment, and that's again part of their role.
Okay. So I think the numbers are something like 1.75 million people in the US suffer a traumatic brain injury every year. And there are like 800,000 stroke victims and maybe somewhere between 4 and 10 million concussions. So, given the large numbers of people with these kinds of brain injuries every year, it seems like it would be a very active area for research and development. What's limited the research in the past?
Yeah, it's very sobering. I mean, basically, stroke is the number two leading cause of death in the world, and traumatic brain injuries are the leading cause of death in younger adults. Both are leading causes of disability. So you would think that there would be, tremendous amount of research here, given the amount of, disability and death that can be prevented. And unfortunately, it's not. It's an area that's significantly under-researched to most other areas in neuroscience and in health in general. Uh, there's probably two main limitations that I would highlight that really probably have held the area back. The first one is that in terms of animal models, you think that injuring an animal model would be similar to injuring a human brain or the way it responds. But most early research is done in mice and rats. And the brain of a mouse or a rat is quite resilient. It looks a lot different. It's smooth. It's, smaller, a lot more simple in terms of its function. And the rodent brains can be a little bit misleading. You give them kind of a little bit of a push or a nudge with, something that might be helpful, and they can do quite well. They recover much better. So there's been a lot of things tested over the years in mice and rats that look like they have a lot of potential. But when you put them into a larger brain, a human brain, uh, they don't perform as well. And so it has been misleading in terms of those early studies. And so not having a great translational model has held the field back or led to some false starts. And then the second big thing I would highlight is the clinical trials that need to be conducted to prove whether or not any intervention, any drug works have been challenging. And the reason for that is that the injuries are very heterogeneous. You think about a stroke, it can happen, in a large vessel and a small vessel in the cortex, the front area or back area of the brain. By the time that patient comes in, it could have been, already, blocked, or that vessel could have been blocked for, 12 hours, it could have been blocked for 1 hour. There's just a lot of different variations. And so you look at stroke trials over the years, people that would come in that were, unconscious and you think were, very unlikely to, make it through, uh, can be better the next day, as, you get some just spontaneous dislodging of the clot. And then other people that are still conscious, complaining of a headache, walking in, maybe dead in 24 hours, and regardless of what you do with your intervention. So the trials have been really challenging, especially in the 1990s and 2000s, where they're taking pretty much all comers for a stroke or all comers from a head trauma, and that variability in how these people had outcomes really clouded any chance that any medicine or approach would have. So both those are areas that you can address, but it has definitely limited research over the years.
Okay. So what did Dr. Lecklider's early research with astrocytes demonstrate? What was the and kind of what was the key innovation that qualified for patent protection?
Yeah, so one thing that he was doing is actually, interestingly, he was originally trying to research calcium signaling in these glia cells and these astrocytes as kind of a different approach. He was actually trying to kill the astrocytes. He was hoping that with certain molecules that trigger these receptors, initiate a calcium cascade. Calcium cascades are usually, bad for a lot of cell types. And if you could kill the astrocytes, this could be a treatment for cancer. There's some glioblastomas, brain cancers that are out there. And when he was trying these different molecules, surprisingly, it was doing what they were supposed to do in terms of inducing a calcium cascade and some of those signals, but the astrocytes loved it. They were thriving in these cultures, not dying. And then not only could they thrive, when you had co-cultures about tissues of both astrocytes and neurons, it now protected the neurons too. So it was a pretty dramatic effect and definitely opposite of what he was originally thinking. And that original insight is what led to, this, a new patent in the space that would be a foundational patent for eventually for astrocyte. But when he tried doing this, and not only was he able to protect the cultures, he could show in mice that are having strokes or mice that are having traumatic brain injuries, kind of a wide range of experience with these mice, that it was consistently neuroprotective, and there was less damage from any of these insults. So it was exciting, it was consistent, it was a novel approach, and yeah, something that we thought could demanded further research.
So given that other neuroprotective candidates had been advanced and failed, and uh, what did you learn from those failures in other programs? And what have you done differently?
So, like I said, one of those big challenges in this field is that the mice and rats might be misleading. And when we talk to investors, when we talk to grant review committees, or people that are experienced with that, a lot of them just kind of would highlight, the lack of translation in the past. So what we really was fairly obvious what we needed to do was prove that this drug works in a larger brain, in bigger animals. So we proceeded to move forward with a series of studies, one where we looked at, traumatic brain injury in a pig model, and then also a transient stroke that would happen in a non-human primate, in a monkey model. And the thing with pigs and monkeys is they have much larger brains. Their brains look like human brains. They're geniocephalic with the folds. They have gray and white matter. So the way that they respond to an injury is much more like how the human brain responds to these injuries. So we went into those types of studies, which would be generally better translational potential, but more convincing for everyone else, adjacent to the company. And we had great results in these pig models. We were able to reduce the amount of whole brain edema, that brain swelling after a trauma, by close to 30%. And in the monkeys that were having these strokes, when we administer our drug, we were able to slow the growth of these, of the lesions that are occurring, as they grow over time when you're having a stroke. We were slowing that by 50, 60%. We made these infarcts be 45% of the size that they would have been if they were not treated. So, great results in these large brains. And that was just much more convincing to investors or grant committees and everybody else around it that this looks real. This looks exciting. This is something we need to move into human patients.
In terms of stroke, does it work in both ischemic and hemorrhagic stroke, or how soon does it have to be given? I guess there's a time limit usually, right?
Yeah, so most people are familiar with the really the only treatment out there for stroke are treatments that help kind of open up the pipes again. If you have a clot in in your brain vessels, blood vessels, you want to remove that clot, and you can do it by a drug called TPA, Alteplase. It helps chew up the clot. Or surgery. That's called an endovascular thrombectomy, where they go in and actually can poke it with a basket and grab the clot. Both of those treatments, those interventions, can only be done if there's a clot. That it's an ischemic stroke. You can't do those in the other type, the hemorrhagic stroke, where there's a broken blood vessel, it's bleeding. TPA in particular, you don't want to administer an anti-clotting agent to someone who's having a bleeding stroke. It would make it worse. So for both of those, you have to wait until you get the patient to the hospital, have them imaged, typically in a CT scanner, and confirm there's a clot. And then and only then can you now proceed with doing a TPA treatment or the endovascular thrombectomy. And with TPA, there is that time window too. You have to do it within 4.5 hours, otherwise the risks outweigh the benefits. So that's kind of the landscape that currently exists. For us, well, we have the benefit where we see advantage in both the ischemic and the hemorrhagic stroke, and in traumatic brain injury and trauma and other hemorrhagic, uh, injuries. So when we, are treating these patients, because we're just going after the blood tissue, we're not going after, or the brain tissue, we're not going after the clots. Uh, we could actually treat earlier. We don't have to wait for that imaging to be treating. We expect that this eventually, our vision was that this would be in the ambulance. When you have emergency responders, they recognize it's a stroke or it's just a head injury, you're able to start administering this drug quickly, and that will allow more brain to be saved. But at the same time, we don't have that safety liability like TPA at 4.5 hours. We see some benefits as late as 24 hours. If it's a slow progressing stroke, sometimes these can grow over 24 or 48 hours. We've treated some animal models as late as 24 hours, and we still see a benefit. So it's a pretty broad window where we can treat earlier and later than what's currently in the clinic.
Wow. So I think you recently completed a phase 1 on AST-004. Can you say more about the results of the trials and side effects, if any, all that sort of thing?
Yeah, yes, we're very pleased with the safety profile of AST-004. So we've done 2 phase 1 safety studies looking in just healthy, normal people. What is that safety profile of the drug at the doses that we want to use in patients eventually? And in all the subjects here, the most common adverse event that we saw was people mentioning headaches. We see that in the placebo group as well. So no major safety signals, no significant adverse effects, which is fantastic. And you could probably still have some of those adverse effects, and you still probably give it to a person who's having a serious stroke or trauma. But the fact that it has a really clean safety profile so far shows that we could probably be doing this also in people that are having more mild traumas like concussions. If someone has concussion and you can take this medicine that has a very safe profile and a potential to reduce the swelling and limit the amount of symptoms you're having over the next few days or weeks. You're more likely to administer that to a child having that football tackle concussion versus if it did have a lot of potential safety issues. So we're very happy that progressed. The trials were successful. We confirmed that the drug gets across the blood-brain barrier and it gets into the CSF where site of action would be. So it was very successful, and now we're ready to move to phase 2 clinical trials.
Yeah, and so when do you think that phase 2 might begin, and has the FDA approved the protocols already, or where?
Yes, so we've submitted everything from our preclinical and phase 1 work to the FDA. They've approved the IND, the investigation new drug application. They approved the phase 2 protocol. That is what we've planned to do in the United States here with stroke patients. So that has been reviewed and approved. We are currently working with several sites and also with the NIH on the plans. And we hope to start this during 2025. So if people are having strokes in 2025 at these various hospitals and sites, there's a chance that they could be enrolled in our clinical trial and be administered AST-004, and hopefully we'll see a benefit here in treating those stroke patients.
Oh, great, great. So I noticed that you have just a really prominent list of stroke researchers on your team, and they've all got significant clinical trial experience. Can you tell us about them and how they came to be involved in the company?
So yeah, so that was one thing that we knew early on, that given the challenges with clinical trials in this space, it is critically important to have the top experts, the top thought leaders in this space actively involved and working with the company on the clinical trial design, not only to make sure that we have the right design that's as optimal as possible, but to also, reassure and validate other people, their investors, other people that might be joining the company, other people that might be joining the trial, that this is, a credible, serious effort. And so we did. We went out and looked for, who are the top people, the top voices in this space. And people that publish, people that lead the committees, the people that, at conferences are the ones that other people really listen to and have had success. And so we approached people like Dr. Jeff Saver at UCLA, who we think is the foremost thought leader in stroke and has been involved in most of the major clinical successes in the world of stroke over the past, 20 some years. And when you approach people like that and show how serious we are, that we're taking a large pharma approach to this in terms of really characterizing and de-risking the asset, we've done the large animal studies, have impressive data. They can tell that we were, doing this the right way. We're doing this serious. We're looking at, very intently at the PK and the dosing and a lot of those characterization aspects of the program. And so they were excited to join up with the company and be part of the clinical advisory board. And we did. We looked for the top stroke imaging expert out there, and we're able to get Dr. Greg Albers involved, the top stroke neurointerventionalist we think in the world, in Raul Noguera, and then someone who's really knows the pharma space, recent clinical trials, a rising star, and Dr. Kevin Sheff at Yale. So we're very, very excited to have such a prominent clinical advisory board across the stroke area, and also in traumatic brain injury. Dr. David Wright, who's led some of the largest, most complex clinical trials in traumatic brain injury. So that again, it speaks, I think, to the credibility of the program. That it's just that we have great data. It's very promising. But these are people that know the space, and given the challenges, we need to design this trial the right way to see if it is actually successful.
So, concussions are all over the place, all over the news, right? And do you see AST 004? Do you see that being utilized eventually by athletes? What about the NFL,
Yeah.
soccer, that sort of thing?
Yeah, we've actually been working with the Department of Defense on this as well. They've been impressed with our data. Traumatic brain injury concussions are one of the top injuries for the Department of Defense. And so they helped us fund some work to make oral disintegrating tablets, versions of AST-004, because that's what would be used on those sidelines of those athletic fields, but also in those military soldiers' backpacks. You can imagine little blister packs full of these tablets. And then, so when that NFL player, when that high school athlete has a bad concussion, they're on the sideline. Hopefully, they're being removed from the game for and going to be put on a rest and concussion protocol. But you can imagine taking one of these tablets, and we've just seen with the AST-004 that we're able to reduce that brain swelling, really reduce the symptoms. We've actually seen some benefits in terms of long-term effects as well. And so we're preparing a phase 2 clinical study right now in athlete concussions.
Oh,
And one thing that's a challenge in those types of studies is finding people that aren't having a lot of repetitive head injuries, like our NFL football. Unfortunately, these people are hitting their heads multiple times per game. So you would expect their blood to have maybe some brain proteins, some biomarkers that are already elevated. So we are planning a study in Australia with Australian rules football players, which will be pretty exciting. Just these are individuals. The sport is one that's more continuous play, more wide open, no helmets. They're not hitting their heads at all, really.
like rugby, right? It kind of,
It's not rugby.
it's sort of,
It's similar, yeah.
okay.
But it is Australian rules football. And so it does look a little bit like our football, but again, no helmets, more continuous play. But there's still, high speed collisions. And when these people have collisions, we would then be able to, treat them, and any increase in blood biomarkers, any symptoms, any benefits, we know are associated with that particular concussion. So we hope to start the study in April next year in Australia.
Oh,
And if this is successful in treating those athletes with concussions, we see that expanding to a lot of other areas where you could expect
sure,
concussions to occur.
sure. One thing that I thought was really interesting as I took a look at the company and the website and all that sort of thing was that you had received some grant funding from the NIH and the Alzheimer's Foundation because you've shown in mouse models of Alzheimer's that treatment with AST 004 substantially reduced beta amyloid. Can you tell us more about that?
Yeah, so we're very excited about the mechanism here. We think it's a very central neuroprotective mechanism. The kind of the fundamental, kind of the simplest way to think about it is that there are, a lot of different ways to regulate brain metabolism. But
Mhm.
as adenosine triphosphate ATP breaks down into adenosine, those ratios of ATP to adenosine trigger different effects. And when you deplete energy, adenosine levels spike up. And our drug is a mimic of that signal, that adenosine response. And so we think that is, fundamental, a pathway that the brain not only regulates brain metabolism, but also is fundamental to their injury response. So when we are using our drug on this fairly central mechanism, we see lots of benefit in the acute space, trauma and stroke. But if we can give this chronically over a long period of time, we think that there's a lot of other areas where the brain is under stress or challenge and that there can be benefit. And so we've seen benefits in multiple models, and one of those is Alzheimer's. So we've now treated mice that are kind of a beta amyloid model and also ones that are a tau based model. And we see significant benefit in both. So in mice that are, more of a beta amyloid model, we treat them, for months at a time, from month 3 to month 9. We see two-thirds less beta amyloid in the
Wow.
brain at the end of that study. So we know that there's something, central going on here with, helping the glia cells and the brain health and that energy metabolism just be better over that time period. So it's a much longer study, and we're still investigating that with some academic collaborations. But it just speaks again to the upside to some other areas that are beyond stroke and TBI, where the brain is under stress, and we think that there can be benefit.
Will you take that to larger animals next, as you
So
did?
for that one, mice are typically the ones that you use for a longer set
Okay.
of studies. Beyond that, then it gets to be very, very long. So for that one, basically you move almost directly from mice into long-term safety studies and long-term people
Okay.
studies.
Okay. Okay. So, but once you get past strokes and concussions, um, for trials like things like TBI and Alzheimer's, they get really expensive. So, how do you think about the long-term future for astrocyte?
Yeah, so we think about it in two stages. The first stage is a little bit more just prove it works. Get to proof of concept. It's an area where a lot of things have failed before. So let's see that this works. It works in animals. It definitely helps protect the brain. Does it protect a human brain after that stroke or concussion? And you try to design those studies to be as homogeneous as possible. You're focusing on that question, does the drug work? And that's what we're doing. We're doing this first stage, proof of concept, does it work? Once you show it works, that's a very large de-risking event. That's if the drug works in human patients and protects the brain, now you go to the next stage, which is how many other areas does this work? Where else how broad can you go? And so for us, we're looking our first stroke trial being large vessel stroke patients that are also getting a thrombectomy surgery. We would expand that to mid-sized vessels, to pre-hospital stroke, to hemorrhagic stroke patients, and then you'd be looking in trauma, not just in concussions. You'd be looking at mild to moderate TBI, severe TBI. So there's a lot of ways you can expand the label here and indication. But if you get through that first stage and have that big de-risking event, it changes the economics going forward. It's something where it's a lot higher probability of success, and there's a lot more investment potential. So at that point, the traditional route is for a company then to move towards an IPO to fund those larger trials, or work with a larger pharma partner
Mhm.
that is very experienced in running multiple phase 2, phase 3s, and they're excited to fund those as well and and share in the upside.
Right, right. What's the initial commercialization strategy?
So the initial stage here is really looking at that first, proof of concept. Let's see if we can get this out there and treating stroke patients as soon as possible.
Stroke. Okay.
Um, so if we can get you know, as an adjunct to those EVT, any patients coming in with a stroke, how quickly we can treat them, reduce that growth of that lesion, reduce the amount of disability from those strokes. That's where we want to, we see the quickest path to market. So we would do that and then be expanding that very quickly to those other populations. We see a lot of potential for that oral tablet and concussion. It's a little bit further behind, but essentially, we would love to see that in broad use as well.
What are you thinking in terms of duration of the phase two? How long will that be?
So the stroke trial is probably 2 years, depending on the final size. We're still kind of altering the statistics on that. Right now, it looks like about a 2-year study and about 200 patients, but we could expand that as well.
Okay, okay. So you've been at it for 10 years. What do you think is the biggest challenge in terms of moving the research forward, Bill? What's the
Yeah, so I think for most companies, there's kind of a couple answers. One is typically the science, and we've been very fortunate. The drug continues to just show great properties, great potential. It's worked in pretty much every kind of different model that we've put it in. So the science is just, we've been fortunate that it's continued to go forward very nicely. I think the other area a lot of the companies will answer with is funding. And this has just been an area that has seen less funding historically than Alzheimer's or Parkinson's or cancer or other areas. And so there's just less pharmaceuticals companies in the space. There's less venture capital in the space. So for us, I think it's just been just a longer journey that where we had to, get some investor investment to get through a set of experiments that shows positive data, get some grants based on that, and that gets us, another set of data, get some more investment. But it's been a slower process. We think that we've been able to do that, move stepwise and keep a high quality in the program and do it the right way. But it is a challenge. Like I said, we'd love to be in a space where people are making a lot more bets and throwing a lot more money at good ideas. But that's probably the biggest challenge in this space. But now that we've moved pretty far along, now we're at the spot where I think we're a pretty promising bet.
I'd like to circle back for a second now as we kind of close. You've got experience at Pfizer and in consulting that is pretty unusual for a founder and particularly a founder with a PhD. Is there some way that you've been able to apply that experience in a way that might be different from another founder without that kind of background, the way that they might think about a problem versus the way that you, with your experience, would now?
I think it was a great preparation to be able to have kind of that consulting background, that Pfizer background. It gave me a broad view of kind of the industry, the major segments and players. Really understood a lot about how drug development works, all the different stages, all the different disciplines that are involved, sub-disciplines. So I felt like I had a good roadmap and landscape, which is probably a little bit atypical. Um, I've also been very fortunate to have a very good network. So Pfizer, for example, is a great place with a lot of turnover as well. So I was able to work there for 10 years in the senior ranks. And I've saw many people kind of come and go, and a lot of people end up in the biotech industry and have other partners. So I think my background was particularly well suited to leverage that network. And that's something that I think is absolutely critical in the biotech space is to have a build your network, find the people that know your disease area, find the lawyers that can be helpful, find the accountants that know the space. So if you don't have that type of background, you got to, organically help, build that. But that was something that I think, I was more the business guy that knew science than just the scientific founder at Astrocyte.
If you are tethered to the bench and perhaps a more withdrawn sort of personality, do you have any advice for that founder in terms of developing his network or her network, as the case may be?
Yeah, I think the traditional scientist, phenotype is a little more introverted and driven into the science. I fit that as well. Uh, but if you want to be in a kind of the business space, you definitely need to think of it as a project that you need to grow your network. You need to educate yourself. You need to participate in, if you're whatever area of the country you're in. There's usually biotech clubs and biotech networks and various weekly seminars that the companies or the lawyers or various people are putting on, and you need to go to those and attend those and network. In this day and age now with Zoom, basically there's so many other seminars and educational things that are happening across the country that you can easily participate in. So I know I get multiple solicitations every week, and I think as any individual wants to educate themselves and subscribes to the fierce biotechs or the
Mhm.
endpoint news or biospace and
Mhm.
follows the news, you're going to see more and more opportunities to learn what's going on in the space. So there's ample opportunity to learn more, and you have to make the effort to take it as a project to grow that network.
Yeah, yeah, and you have to read all those newsletters. I think I get 5 fierce newsletters a day, something. I don't know what it is. Where do you see Astrocyte and AST-004 in 5 years or so?
Yeah, so we're finally at that point where we have a line of sight to these critical phase twos, that this proof of concept. And so we're excited. In the next, I think, 2, 3 years, we're going to finally know if this drug works in human patients. And what's interesting is that this molecule either works or it doesn't work. And ever since it's ever been synthesized, it's got its properties that, is effective or not effective. And we're slowly unveiling whether we know whether that's true or not. And in 2, 3 years, we'll know. So we're excited to see that. If it works, this is exciting for healthcare. It's exciting for many patients that are, suffering these types of injuries. We see the drug is being used in more clinical trials and hopefully then being used more broadly in the medical space. So in 5 years, this could be, gaining momentum, approved and used in some hospitals already for things like stroke and growing. Or the opposite side, flip this coin essentially. And if the drug doesn't work and there's just something unique about human brains that, is different, that doesn't cause this to, have that same level of efficacy, then we're probably, closing shop. This is, a little bit of a binary situation at the end of the day.
So I wish you all the luck in the world, and I thank you so much for your time, and sharing your insights with us today about astrocytes. Thank you, Bill.
Yep. Tim, thank you very much.
Thanks for tuning in to the Life Sciences and Biotech podcast. We'll see you in the next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Doherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Doherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Doherty makes no representation that the information contained in this material is accurate and is under no obligation to update this information as changes occur.